friends, welcome back to my channel Xgene and proteinX. In this tutorial video, I'll discuss about the overview of the lactose permease. So the lactose permease is one of the protein that encodes by the lac-Y gene in the E.coli. So in E.coli, in the lactopyran, it has three different genes, lac-G, lac-Y and lac-A. But the lac-Y, it solely encodes lactose permease. And the lactose permease is responsible for the transportation of the beta-galactosides into the cells. The beta-galactosides are the compound that contain the beta-galactoside linkage. So most abundant beta-galactoside compounds is lactose. Lactose is utilized by the lactose is utilized by the bacteria for their energy source, and the lactose permease it transport this lactose into the bacterial cell by exploiting different kind of mechanism, that is the symport system and that is the proton gradient system. And the lactose permease belongs to the major facilitated or superfamily. This is one of the transportation protein transport family, and these are unlike the similar kind of mechanism like glucose transporter. Also, this lactose permease, it has around 417 amino acid residues, and the N terminal and the C terminal, both the terminals, it are located in the cytoplasmic site of the membrane. It also has around 12 transmembrane helices. So as you see in the figure, in the left side crystal structure figure, that each color it representing each transmembrane helices. So right. So the important function that I already mentioned here, the important function of this this lactose permease, that it has been already observed that this single gene product are solely responsible for all of the translocation reaction catalyzed by the galactoside transport system in E. coli. So the transportation system that it exploit, that is very much important. So I'm not discussing very in detailed transportation process here, but just very initial basic part I would like to mention here. So if you're gonna would like to know that kind of transportation system in detail, so you can go through the paper that I cited here. So I already mentioned in the later slides, so you can go through that. So here is the transportation process of the lac-Y. So the mainly it exploits that the lactose H and proton symport. So this is one of the important transportation system that lac-Y utilizes. So lac-Y utilizes the free energy that released from the downhill translocation of the proton, and this energy they utilize for driving the accumulation of the galactosides against a concentration gradient. So this is the main theory, this is the main basic part of this transportation process. So if you would like to know in details, so you can go through that paper. So in my next slide, I'd like to mention the structure or the amino acid residues of these proteins, these lac-Y proteins, this lac-Y lac permease. So this is the amino acid structure, amino acid residues of these lac-Y proteins, and here the researchers they did some point mutation in some parts, and they found that there are some active amino acid residues. These amino acid residues are responsible for some activities like binding and the translocation. So the substrate binding, so there are main two features that are the substrate binding. One is substrate binding and another one is proton translocation. So because the lactose transportation system it depends on the proton translocation. So here I I marked red, so this kind of proteins, these are responsible for substrate binding as well as the proton translocation. So here you can, this amino acid residues I found from the Uniport system. So if you go through this kind of this amino acid residues, so you can identify this amino acid residues here. As well as also there are some this kind of 3 other proteins like arginine, histidine and glucose. These kind of proteins, these are responsible for the proton translocation system. Alright, so here the helix, these helix, these are representing that in which part this amino acid residues are locating. So in my next slides I'm gonna show you the crystal structure of these proteins. So here it's showing the crystal structure of the amino acid of these proteins, lactose permease, and here it is the binding that beta-galactosides. So how it binds, so this is the crystal structure, but the similar kind of structure it is representing here by the schematic diagram. So here each color of the crystal structure, it's showing each transmembrane. So in total around 12 transmembrane helices here, and here you see the 6 transmembranes. These 6 transmembrane helices are here in the N-terminal part that are representing in the blue color, and also the C-terminal. The C-terminal in this part, this is in the C-terminal here in this part. So C-terminal are in the red color. So this is also in this part also there are 12 transmembrane helices. But here is, so if you take a closer look, so you can see here there are green color some amino acid residues. So these green color amino acid residues are responsible for the substrate binding. So as I mentioned already in my previous slide, that which amino acid residues are responsible for the substrate binding. So here you see there are around 1, 2, 3, 4, 5, 6. So 6 amino acid residues are responsible for that. And also the yellow color amino acid residues, these are responsible for the proton translocation. So these all amino acid residues are located in the C-terminal part. Alright, and also here in additionally here another amino acid residues, these are responsible for the both job like substrate binding as well as the proton translocation. So here in this paper they use one of the chemical compound, that is the TDG. This is one of the chemical compound. This is one of the beta-galactoside compound, and here they found that how it bind, how this lactose permease it bind with these beta-galactosides. So this is how, like in the N-terminal and in the C-terminal, in between there is one hydrophilic cavity, and in this hydrophilic cavity this compound, this beta-galactoside, it binds. So this is how it works, and this is very much important transportation system of the beta-galactoside into the E.coli cells. Alright, so here is that paper, that the reference that I added here. So this is one of these important paper. So if you wanna check, that's you can go through also. Alright, so thanks guys. So I hope this video will be helpful. If you like this video, kindly hit the like button, subscribe my channel and share this video. And if you have any queries, kindly write in the comment section. Thanks.